Welcome back to the lab. We've spent so much time talking about genes and molecules, but what if we want to get our hands on the single perfect cell we need for our research? Today, we're diving into a technique that sounds like science fiction, but is a cornerstone of modern biology, FACS, or fluorescence-activated cell sorting. We'll show you how we go from a messy biological sample to isolating a single perfect cell. And we're back. At its core, FACS is the process of physically separating a mix of cells into different populations based on their unique characteristics. Imagine a high-tech detective agency that can spot a single suspect in a massive crowd. This is how we isolate rare stem cells, study how our immune system fights off diseases, and even diagnose illnesses. It's the ultimate tool for getting the right cells into the right place for our experiments. So how do we actually do this? It's a three-step process that's both a delicate art and a precise science. Step 1. Get your cells ready. First, we have to prepare the sample. You can't just throw a piece of tissue into the machine. We need to break it down into a clean, single-cell suspension. This often involves gentle enzymatic digestion to break apart the tissue, followed by filtration to get rid of any clumps or debris that could clog the machine. Step 2. Put on the flags. This is where the magic happens. We use specific fluorescent dyes or antibodies to tag the cells we're interested in. Think of it like putting a tiny, unique colored flag on every single cell. We have to be careful with this step because a dim signal or low marker expression can ruin the whole experiment. Step 3. The high-tech sorting. Now the cells run through the machine one by one. As each cell passes a laser, the laser reads the color of its flag. Based on that color, the machine gives the cell a tiny electric charge and then uses a magnetic field to divert it into a separate tube. It's like a high-speed traffic director for cells, sorting thousands of them per second. As with any advanced lab technique, things can and will go wrong, but a good scientist knows how to troubleshoot. Problem 1. You didn't get enough cells. Sometimes your cell yield is just too low. The solution? Check your tissue digestion and minimize handling time to keep the cells healthy. Problem 2. My machine is clogged. This is a classic. Cell clumping and debris are a frequent problem, so always be sure to properly filter your sample and use gentle pipetting techniques. Problem 3. My cells are dying. Low cell viability is a major issue. Remember to keep your samples cold and avoid overdigestion of the tissue during preparation. By following these simple steps, you can ensure your results are reliable and reproducible. A FACS is a testament to the incredible precision of modern biotechnology. By isolating specific cell populations, it opens up a world of possibilities for genetic and cellular research, from diagnosing diseases to understanding how our bodies work at the most fundamental level. Thank you for joining me on this deep dive into the FACS technique. If you enjoyed this video, please give it a thumbs up, leave a comment below with your questions, and subscribe to the Geneticist for more episodes on how genetics is changing our world. See you soon.